So here is a typical picture of a conventional library. So if you were to define what is a library in conventional sense, so it is a sizable collection of books or records which are sorted or ordered in a certain manner, and this library should be searchable, right? You should be able to retrieve the book of your interest following an index. So a library in biotechnology represents a collection of related but different nucleic acid fragments ligated into a vector, transformed into a host cells, and maintained. One is as plate cultures, so you grow them on plates, and the other is as glycerol stocks, which can be stored for a longer time and can be retrieved as and when required. Typically, there are two types of libraries in biotechnology. Number one, what is known as the genomic library. This is generally prepared for genome sequencing projects, also sometimes for DNA fingerprinting, and we'll talk more of it as we go along. And the other important type of library basically is the cDNA library, or the complementary DNA library, which is basically derived from RNA, right? And is utilized for transcriptome analysis. So this is how a typical genomic DNA library would look in a lab, right? So here you are. This is a typical Petri plate with culture medium, and these are different types of colonies that are growing. Of course, they would all be of the same color, but to indicate that each of these contains a different genomic fragment, I give them different colors. So let's now move on and talk of genomic DNA library first, and then come to cDNA library. So here we are, and we want to basically make a genomic library of the DNA present in these cells here, right? So this is your eukaryotic cell. You have a plasma membrane, you have a nuclear membrane, and inside the nucleus, you have the genomic DNA. So the first step, of course, is to extract the genomic DNA. That could be done using phenol chloroform method or triazole method, and then you need to fragment it to generate smaller fragments, right? So this is some of the fragments that you generate. These fragments can then be ligated into vectors, and vectors can be transformed into host cells, giving you the final library here. So here I show you three cells that contain different fragments coming from the original genomic DNA here. So a genomic library is a collection of uniformly sized overlapping DNA fragments such that every part of the genome is represented at least once in the library. So there are two important points to remember here. One, generally the library is uniformly sized in the sense that you select for certain fragments of specific sizes to be included in a library. Because you create a genomic library for sequencing projects, the overlap between DNA fragments is important. Why do you want to have uniformly sized fragments, and why they should be overlapping is something that we'll talk about as we move along. Now let's look at the process of genomic DNA library preparation in more details. So here are the host cells, and you want to prepare a genomic library of the DNA present in these host cells. So the first important thing is the extraction of DNA, which can be done by either phenol chloroform method or the triazole method. And then the second important step here is your generation of overlapping fragments. And to do that, you can do a partial additional restriction enzymes. You could do physical sharing, or you could do sonication, in which case you give high frequency sound waves, and that also results in random breakdown of DNA fragments. So after your fragmentation of DNA, the DNA has to be size separated so as to ensure that you pick up a DNA of a specific size, right? So for that, what you do is agrogel electrophoresis. So here you are, you're doing the size separation using agrogel electrophoresis. These are the wells where you load your sample and then do your electrophoresis and allow the DNA fragments to separate out. So the gel is cut at parts which correspond to the size that we want to take forward, and then the DNA in this gel is eluted using elution kits. Next, you have to ligate these fragments into vectors. So you do your ligation reaction, and then once the ligation is done, you do your transformation reaction. Finally, you get a collection of host cells that contain individual fragments coming from your original genomic DNA. So like a conventional library now, this is a sizable collection of DNA fragments from an original genomic DNA, and the library can be searched, and the required fragment can be retrieved from the library for downstream use. Mostly this would be for sequencing. So then, in order to construct a genomic DNA library, the organism's DNA is extracted from cells and then digested, shared, or sonicated to generate overlapping DNA fragments, and finally size separated to obtain fragments of a specific size. So each library would have a specific range of fragments sizes that it would contain. So once you get to fragments, you do your size separation and pull out the specific size that is required for your library preparation. So the size of the fragment that you want to pull out from here would depend on the sequencing technology that you're using. If you're using Sanger sequencing, you know that Sanger can read 900 bases starting from one end. So around 2 KB of fragments can be accommodated there. So in order to construct a genomic library, number one, the organism's DNA is extracted. So this is here, you extract the DNA, then you digest, shear, or sonicate the DNA to generate fragments. You get it here. And then, of course, these fragments are size separated on an agarose gel so as to facilitate the user to pull out fragments of a specific size range, right? So to generate overlapping DNA fragments and size separated to obtain fragments of a specific size, right? And because you're doing random fragmentation, what you get is actually overlapping DNA fragments, which is very important in case of genome sequencing because that would help you assemble the genome, and we'll discuss this in more details as we go along, right? So then the fragments are then ligated to vectors, and vectors are then transformed into the host cells. Commonly a population of E. coli or yeast can be used, and each cell contains one fragment of the genomic DNA that you began with, right? So this is your library here. And using a host cell to carry the vector allows for easy amplification and retrieval of the specific clone from the library for downstream analysis. So this is the process here of making a genomic DNA library. You need to first extract the DNA, you need to fragment it, you need to size separate to allow only a specific size fragments to be cloned into your library. Then you'll get into vector, and vector is then transformed into the host cell. And this library can now be grown as a plate in a nutritive media, or it can be kept as cholesterol stock for long-term usage. So let's say this is your genomic DNA here, and after fragmentation and selection for a specific size range, you get these fragments that are roughly uniformly sized with each other. The other important feature of a library, as I told you, was you need to generate overlapping fragments. So these have overlaps. For example, the 3' end of 1 uniquely overlaps with 5' end of 2. And that allows you to decide the order of fragments also in the assembly. So you know that 2 follows 1. Likewise, the 3' end of 2 uniquely overlaps with 5' end of 3, which means 3 follows 2, and so on and so forth. So for the purpose of our convenience, we say that all of these pairs of fragments have unique overlaps with each other, and therefore, once you have the sequence of these fragments, you will be able to assemble them very easily, right? So now, if you see, you make a genomic library out of this, and you could have a complete library or you could have an incomplete library. So in case of a complete genomic DNA library, every single fragment that you have here is represented at least once in your library here. So a complete genomic DNA library would conform to all features of a genomic DNA library. For example, here, it is a collection of uniformly sized overlapping DNA fragments such that every part of the genome is represented at least once in the library, right? What can also happen at times is that the library is incomplete, and this could happen due to several reasons, one of which could be the incompatibility of certain sequences to be ligated to certain vectors. So in this case, for example, here, you could have a case where fragments 4, 5, and 9 are not compatible to the vector, and therefore they're not ligated, and they're also not then transformed. So in such case, the library that you get is an incomplete library. You will not have representation of these fragments, and therefore the genome sequencing that you do for this would also be incomplete. And to complete the genome sequence and to complete the genome assembly, you'll have to make the library again with a different vector.